Thank you, Peter. Yes, I was co-chair of the Biogeochemistry Working Group for a little over 10 years, but now that's been handed off to the local co-chairs, younger scientist, Matt Long, get some fresh blood in there and better new ideas besides us old fogies. So as Peter said, I'm going to be talking about ocean biogeochemical modeling. And I'm also going to talk a little bit about the coupled carbon cycle in CESM in other climate models as well. What do I do to advance? There we go. Thank you. A brief outline of the material that I'm going to present here. A handful of topics that some I'll go a little bit more lightly into because of time constraints, but first I'm going to talk about some of the large-scale ocean biogeochemical features that we endeavor to get the processes correct in our models so that we can reproduce in our models the observed features of biogeochemical properties. I'll talk about some of the techniques that we use for modeling the processes that are highlighted from the observations, talk briefly about how we assess these models, and then talk briefly about the global carbon cycle in CESM and other climate models. So I'm going to start with observations because doing models of the Earth system is not something you do just abstractly where you just make things up. We want to reproduce the world that we live in. And so that starts with observations. And what we want to do is have our models reproduce to the all extent possible what the major features that are observed in reality. So I've got a couple figures here that are from ship tracks going across the Pacific Ocean from Antarctica up to the Aleutian. See, do I have a point? Yeah, here we go. So these are starting at the Antarctica, and this ship track goes all the way north up to the Aleutian Islands. And this top figure is, so latitude is going left to right, and depth is going top to down, and the top panel is nitrate, and the bottom panel is dissolved oxygen. Nitrate is a nutrient that phytoplankton, the critters that grow from sunlight need to support life. It's a major constituent of the biological membranes. It's part of DNA. It's part of the biology, part of the cellular structure, and it's required for life. And oxygen is also required for life, and it's consumed and produced by different biological creatures in the ocean as well as in the land. But these are measurements of these concentrations of these quantities from thousands and thousands of measurements. If you got really close to this picture, you would see lots and lots of teeny tiny dots. Each of those represents a measurement of these quantities in the ocean. And then we've contoured those observations into these color maps. There are a lot of features that are evident in these figures that are there because of different processes in the ocean. And I'm going to go through those and then talk about the processes that lead to those features in the observations, and then how those processes, then we'll talk later about how those processes are represented in models. So the first thing that I'm going to highlight is this nitrate, which is this nutrient. In the surface waters, it's very, very low. And that's because biology has consumed it all, primarily. That biology, photosynthetic life in the near surface waters of the ocean consume nitrate until there's no more left. But they can only grow. They also require light for them to do photosynthesis. These are the critters at the bottom of the food chain, the photosynthesizers, the autotrophs of the ocean. They require light to consume that nitrogen and other nutrients. So this only happens in the near surface, only in the top 100 meters or so of the ocean. And so this nitrate is consumed initially in the cycle of the nutrient. Nitrogen is consumed in the surface waters. In this, the critters convert this into organic matter. So this is their cellular matter. And then they have a life cycle where phytoplankton have a very short lifetime, just days. And after they pass away, and they also get consumed by critters higher up the food chain, zooplankton. And the outcome, they're dead carcasses, whether it's directly from the phytoplankton that decease and then start to sink through the water column. And bacteria in their cellular matter is broken down by bacteria back into, from that organic matter, back into an inorganic form, back into the nitrate. So the nitrate is consumed in the surface waters from photosynthesis. And then as the dead carcass, as the dead detritus sinks through the water column, it's converted back into this inorganic nitrate form. And so one of the things you see here is this is where some of that accumulated respired nitrogen occurs, is places like here, this intermediate depths in the North Pacific, where the nitrogen, nitrogen content of the organic matter remineralizes back into nitrate and leads to a local maximum here at these depths. Now, if those were the only processes going on, you would just expect to see high values and low values, but there's other characteristics going on of this distribution. One is there's this large scale north-south gradient of nitrate in the ocean. And this is due to the large scale circulation of the ocean model. Peter referred to the meridional overturning circulation. So this is this large scale circulation of the ocean. And I think Gustavo probably described this in his previous talk as well. So the large scale circulation of the ocean is also leading to a non-uniform distribution of nitrate in the ocean. When those, I'm going to go on to oxygen. When those critters consume nitrate and convert it into organic matter, they produce oxygen, much like trees that are out here. Photosynthesis is consuming carbon dioxide, consuming nutrients, producing oxygen. So we have elevated values of oxygen where that photosynthesis is happening in the ocean. And then when the bacteria breaks that organic matter down, oxygen is consumed in that process. So much like when soil is remineralized, consuming oxygen, this also happens in the ocean. So there's largely a negative correlation between nutrients and oxygen in the ocean. So where this remineralized nutrient is maximum, the oxygen is minimum, because that remineralization of the organic matter primarily by bacteria is leading to low values of oxygen. And like the large scale circulation is impacting leading to other distributions here, there's a large scale circulation impact on oxygen. But that's not quite all that's going on, because if you look at low latitudes, like close to the coast of Antarctica, there's not much consumption of nitrate going on. There's still lots of nitrogen there. And there's two reasons for that, among others. One is because it's cold. And so the growth happens more slowly in a cold environment than growth in a warm environment. But also there's minor nutrients like iron that are required that are in low supply in the Southern Ocean. So there's another requirements for biological growth that are not present. And so there's less growth in the Southern Ocean than at intermediate latitudes. And because of that, this nitrogen does not get consumed as much. So we have large values near the surface. There's plentiful nutrients that's not getting consumed by biological productivity. However, what I said before about mostly oxygen and nitrogen are negatively correlated. If you look more carefully, you see that oxygen is also high in the Southern Ocean. So we've broken that negative correlation between the nutrient and the oxygen. Even though there's lots of nutrient, where we would expect to see, here's another place where there's lots of nutrient and there's low oxygen. That's not the case in the high latitude Southern Ocean. there's lots of oxygen and lots of nutrients. So there's another process going on. In the case of oxygen, this is air-sea gas exchange of the dissolved gas. So unlike the nutrients, which are primarily just being circulated in the ocean, and they're not crossing the air-sea interface primarily, oxygen as a dissolved gas in the ocean exchanges with the atmosphere until the gas flux tries to restore the concentration of oxygen to a equilibrium solubility, and cold water holds more dissolved gas than warm water. One way to think about why that happens is that in warm water, as the molecules are getting more excited by the heat, it increases the partial pressure of the dissolved gas in the water, and so it can hold less of it in warm water than in cold water. And because of this temperature-dependent solubility that regulates the air-sea gas exchange, the cold waters of the Southern Ocean are rich in oxygen because of that air-sea exchange. It's not because of the biological production of oxygen, it's this temperature-dependent solubility. So these are some of the major features of the major processes that are regulating the distribution of nitrogen and oxygen that we need to include in our models. And that one that we really care about, I haven't mastered this, thank you, is carbon dioxide. So one of the main reasons I do ocean biogeochemistry as Peter referred to, at an altitude of 6,000 feet above sea level, and at a place called the National Center for Atmospheric Research, which you might wonder, well, why do we do that? Well, one of the reasons we do ocean biogeochemistry at the National Center for Atmospheric Research is because the ocean plays such a large role in the regulation of atmospheric CO2 concentrations. And that then plays a large role in regulating the climate, as you are probably all aware. And so the representation of carbon dioxide in seawater is usually referred to as dissolved inorganic carbon. So you can think of dissolved inorganic carbon in the ocean as something like the dissolved CO2 in the ocean. There's a bit more to it because there's some complicated chemistry as the dissolved CO2 interacts with seawater, leading to other carbonate species in the water. But for now, I'm not going to get into that. You can think of it primarily as this is the carbon of the atmosphere represented in the ocean model. And I'm showing here another transect of observations. And this is total dissolved carbon dioxide in the water, plus all of the other carbon species from the chemical reactions. And we see the same sorts of features that we saw for nutrients, that it's low in the surface waters, because when that biological activity leads to production and growth of organic matter, that consumes carbon from the seawater. And that leads to a reduction of concentrations of carbon in the seawater. And then in that sense, DIC looks a lot like the nutrients because it's governed by biological productivity and remineralization of organic matter, just like the nutrients. So we see low values in this low latitude surface waters, and higher values in these intermediate depths where the remineralization from bacteria converts that organic matter back into an inorganic form. However, DIC also has exchange with the atmosphere. This is how the ocean plays a role with regulating the atmospheric concentration of CO2. So DIC has two hats. It looks kind of like the nutrients because of the biological imprint on DIC, but it also has some aspects where it looks like dissolved oxygen because it also has a temperature-dependent solubility with gas exchange. So DIC has multiple processes that regulate the distribution that is observed in the ocean. And we need to include all of those processes in our models so that we can reproduce the observed distributions and then make predictions about what we think will happen in the future with different scenarios. I know that some people like to deal more with words better than with figures. And so I'm going to summarize this I still have not figured out how to advance this. Thank you. Actually, one more slide before I summarize with words, sorry. This air-sea exchange with the ocean. This is a map of the flux of air-sea CO2, also from observations, from shipboard measurements of constant difference between partial pressure in the ocean and partial pressure in the atmosphere of carbon dioxide in computing what the flux would be. This is a spatial map of that flux. There's large-scale outgassing happening in the equatorial Pacific, and this is because that large-scale circulation is bringing carbon-rich cold water up to the surface. Then that water heats up because of the intense heating from the atmosphere. As it heats up, the water cannot hold as much carbon dioxide, and it releases carbon dioxide to the atmosphere. So you have outgassing of carbon dioxide where these cold carbon-rich waters are reaching the surface. You have regions of intermediate latitudes where water is being subducted, and this is carbon is being absorbed by the ocean in these regions. And at high latitudes where you have old carbon-rich water upwelling, thinking of the figures that Peter showed with the overturning circulation, you have, again, carbon-rich waters upwelling at these high latitudes that are releasing carbon there. And this leads to these distributions of air-sea CO2 flux that we want to represent in our ocean model and have all the processes that lead to this. So as I refer to now, here's summarizing what I was just referring to in words. The primary processes governing the distribution of the nutrients like nitrate and dissolved oxygen and carbon are biological productivity, which is in the euphotic zone because it requires light. This consumes nutrients and inorganic carbon, produces organic matter and oxygen. The export of that organic matter out of the euphotic zone from sinking particles, this is detritus or calcium carbonate shells for those critters that make calcium carbonate, circulation of that suspended matter, then that remineralization of organic matter converts things back to the inorganic form. It's sort of the reverse of productivity, consuming oxygen. We have general circulation with the overturning, large-scale overturning of the ocean model, of the ocean. There's also lateral and vertical mixing and temperature-dependent air-sea gas exchange. All of those are leading to these distributions, and we need to include all of these processes in our ocean model so that we can reproduce what's going on and believe that we have a faithful representation. These are the processes that govern the global distribution of nutrients, oxygen, and carbon. There are other processes that have smaller impact globally, but regionally are significant. And I'm just going to touch on these briefly. There is some atmospheric deposition of nutrients that comes in through dust and also from byproducts of combustion sources in lightning. So we get iron and nitrate and phosphorus coming in to some extent from the atmosphere. There's burial of some of these elemental quantities into the sediments as some organic matter hits the seafloor and accumulates in sediments. There's loss of material to the sediments. There's also riverine inputs of when you're close to a river mouth, that can be a very large impact on the local distribution of these biogeochemical tracers in the ocean. Globally, not as much, but locally they can be significant. For nitrogen, there's also nitrogen fixation where some critters have evolved the capability to convert dissolved nitrogen gas, which is usually not usable for biological growth. They convert that into ammonium, which can be used for consumption. And this is leading to biological activity. And there's also a denitrification, which is another important term in the nitrogen budget, which is the consumption of nitrate during remineralization when oxygen is low. When there's not enough oxygen to consume, the critters that do that decomposition consume nitrate instead. So now I'm going to talk about some of the modeling of how we model primary productivity in the ocean. One of the main ways we do that is with a hierarchy of models. of the biology and the different quantities. And one is something referred to as an NPZD model, where N stands for a nutrient, which can be an example of nitrate, ammonium, or other macronutrients like phosphate and silicate, or micronutrients like iron. We have phytoplankton, which are photosynthesizers, which convert nutrients into organic form. We have zooplankton, which eats the phytoplankton. They're grazers. And then we have detritus in this NPZD model, which is the dead matter, which is the outcome of the death of the phytoplankton and zooplankton. A diagram of showing an example of how this works is on the right. This is from a particular model from this Fashion paper in 1990, where there's a phytoplankton pool, which all of these arrows represent flows that have processes that represent them. So phytoplankton grow consuming ammonium and nitrate. They get consumed by detritus, by zooplankton, and they also die, leading to detritus and dissolved organic matter. The zooplankton grow, consuming that phytoplankton. And when the zooplankton die, they produce dissolved organic nitrogen and ammonium. All of these arrows represent different processes in the web of life in the ocean, and we need to include a process in the model for each arrow. And so we need to include, it gets quite complicated because you have to have a parameterization for each of these processes. A very simple model in showing how these things can quickly become is just an NPZ model, where you simplify from that diagram just to 3 pools. But if you represent all of the arrows in that pool, you need to represent things like growth, which this first term is growth, which depends upon a limiting of the nutrients and in light. There's grazing, so the phytoplankton are being grazed by the zooplankton, and there's a parameterization of how much grazing takes place. And there's a loss term, mortality. This is the ODE for the balance of phytoplankton. Similar equation for zooplankton for this model is this, so that grazing goes into the zooplankton, and the zooplankton have a loss term as well. And then you have the resulting ODE for the nutrient in the water. So when the phytoplankton grow, you see that this term is exactly right here, that consumes the nutrient. And this mortality term goes back into the nutrient pool. And some of the grazing goes back into the nutrient pool as well. This is a system of 3 ordinary differential equations. And every loss term has a corresponding gain term, so it is conservative. Total mass in the system is conserved. Now, if you were to count all the free parameters in here in this really, really simple model, there's over a dozen of them. So how do we constrain them? Because there's so many, and we have to decide for that simple model, how do we determine what parameters to use in our model? Some of this is based on observations. So you photosynthesis irradiance curves to see if you apply different amount of light to a phytoplankton, how much it grows. You can figure out what the parameters need to be to fit that curve. We can do a similar thing for nutrient additions in laboratories and in field incubation experiments. Some of these are parameters that are hard to constrain with observations. And so what you do is you try different things in your model until your model reproduces observations of the observed phytoplankton or nutrient distributions. And sometimes we just use what previous models used instead of reinventing it and retuning things from scratch. I mentioned that in addition to these phytoplankton classes, and an NPZD has phytoplankton. In a more complicated model, we would have multiple phytoplankton classes represented, and those are called phytoplankton functional types, PFTs. It's somewhat analogous to what happens in the land model, where you have plant functional types, but we have phytoplankton functional types in the ocean model. And these correspond to different types of critters that have explicit biogeochemical roles, that maybe some consume silicate and some of them don't. They form silicate shells. Some make calcium carbonate shells, the calcifiers, and some don't. And so you would represent those differently because of the different ways that they interact with their environment, the way that you can classify them into how they depend upon temperature or other environmental quantities. There's a whole chain of models with different complexity of phytoplankton functional groups. This naturally leads to, when there's different types of models, well, which one is the best? And you've probably, as you're learning to use CESM, of course, ours is the best model. But one of the key things is that there's no such thing as the best model. And I'm going to give an example. One of my favorite pieces from the literature is where somebody, this Friedrichs paper from 2007, took a dozen different ocean BGC models and evaluated how well they do compared to observations at 2 locations. One is in the equatorial Pacific. That's what the EP stands for in these plots, and the other is the Arabian Sea in the Indian Ocean. What Marjorie Friedrichs did, she took the models and tuned them for each location to see, with all these models of increasing complexity, she tuned them to see, well, how well can it do at each of these locations if I try to modify all the parameters to do closest to observations. What she found is if you look in the equatorial Pacific, you can make the simple models work just as well as you can the complicated models. However, if you tune them, in here in these plots, a high value is bad. So that's a penalty function about how well it's doing compared to observations in the Friedrichs paper. In this lower left panel, what she did is she tried to tune the models to do both well at both locations simultaneously. And when she did that, what she found is that the more complicated models did better at both locations simultaneously than the less complicated models. So there was some benefit with that complexity that wasn't evident when you just looked at one site. And then if you ran the tuning at one site and then ran it at the other site, and this is the reason why you would care about doing something like this, of tuning at both sites simultaneously, is in CESM, we're trying to run the same parameters everywhere around the ocean globally and use the same parameters. So we want to know that our model can work well in many different regimes with one set of parameters. So this would tell us for CESM, we want a more complicated model. And another thing with CESM is that we run it out into the future where we're going to encounter situations that are not present in modern day observations. So what Marjorie did to simulate this is tune the model at one location and then see how well it does at the other location. And what she found is that the more complicated models did better. So for CESM, the lesson is that we want to use the simple model is not going to be sufficient to predict the future or work globally. However, if in your studies, you're just working regionally and just trying to understand things locally, the simple model will be sufficient. So there's no such thing as a best model. It all depends upon what you're trying to do with the model. Okay, thanks, Peter. I'm going to skip some slides here. This goes into a bit more of the detail of the structure of what we have in the CESM ocean biogeochemical model. And I'll do the text version of it, the primary features of the ocean biogeochemistry model is that we represent 4 different classes of nutrients, nitrogen, phosphorus, silica, and iron. We have 4 different phytoplankton functional groups. We're doing a trade-off of having some complexity in the model, but not 10 functional groups or more. We have 3 autotrophs. These are the critters that use light to grow in one grazer. And we have one category where placholithophores. These are the critters that make calcium carbonate shells, the chalk. We don't use chalk that much anymore these days. And I'm a mathematician, so I use chalk. This leads to 32 tracers in our ocean model as of CESM2. In earlier versions of model, we used a slightly less complicated version, which had fewer tracers. Some of the aspects of where we try to simplify the model is that we assume a constant ratio of carbon to nitrogen in these plankton, which is not entirely in alignment with observations, but it simplifies and reduces the number of tracers in our model. We have variable ratios of phosphorus to carbon, iron to carbon, silica to carbon, and chlorophyll to carbon. And there's a sophisticated representation of iron in the model. There certainly are some known gaps in our model, things that we don't represent. The calcification and open ocean dissolution of calcium carbonate is independent of the chemistry of the calcium carbonate. The riverine inputs of our BGC tracers is prescribed instead of prognostic. One day we'll have a coupled model of nutrients coming from a land model that computes prognostically what's going into the ocean, but we're not quite there yet. And we do have a bit of a loss of terms that get buried in the sediment, just get lost from the system. We're not tracking BGC and sea ice, and we're focusing primarily in CESM on lower trophic levels. So the model that's in CESM is primarily focused on the global carbon cycle at the lower trophic levels. There's work ongoing to connect our model to fisheries for future versions of CESM. I'm going to skip over, well, I'll just describe briefly. There's a large one. I started the talk off by showing observations. And so we go back, once we have our model put together, we go back and compare our model to a large variety of observations so we can become convinced that the model is truly representing what is going on in the earth. And that gives us confidence that our predictions for the future are something worth looking at as opposed to just making stuff up. Some of the big challenges of doing this modeling, like with all the components of CESM, is we have many, many parameters in our model, and we need to optimize those to represent the world as we represent the observations. And this is complicated when we have so many parameters in the ocean model, just like it is in the land model, for instance. And there's biases in the climate model that we don't want to overcompensate for with the tuning of the BGC. I'm really short on time, but I will mention that this all is one component of the global carbon cycle, where the carbon is being transferred between the land, the soil, plants and soils, and vegetation, and ocean, and atmosphere. And all of these, the big numbers represent stocks of carbon inventory in these different reservoirs of the Earth carbon system. There's approximately 10 times as much carbon in the ocean as there is in the land, which has many times more than what's in the atmosphere. This large reservoir of carbon in the ocean is then a large control on atmospheric CO2, which is why we do ocean biogeochemistry at the National Center for Atmospheric Research. We've now been doing simulations with coupled carbon climate models in CESM, where we look at prognostically predict how much carbon is going in and out of the different components of the land and ocean into the atmosphere, and also with fossil fuels. And if we compare this to observations, we do reasonably well with recent versions of CESM. These are time series of fluxes into the atmosphere. And the gray bars are observational constraints, and CESM is doing a reasonable job of reconstructing the global carbon cycle fluxes. We do a reasonable job of capturing the seasonal cycle of atmospheric CO2 at various locations around the world. In the top left, we have the seasonal cycle of atmospheric CO2 at Point Barrow that the model is capturing reasonably well as at Mauna Loa and Antarctica in the South Pole. This is putting this a bit into context of other modeling centers around the world that are doing similar experiments with their models. We're not the only ones that do coupled carbon climate experiments. This whole laundry list of modeling centers are doing similar experiments with CMIP, and NCAR is sort of in the middle of the pack in terms of representing the global carbon cycle in terms of skill. There's a whole literature on how the CMIP models are representing the carbon cycle in CMIP 5 and CMIP 6. And I'm mostly listing this here. If this is an interest of you, I'm not going to go through all of these in detail, but primarily listing this because the slides are available to you so that you can dig into the literature and learn about all the exciting science that's going on to constrain the carbon cycle and Earth system models and what we're learning from it. So wrapping things up, there are large-scale ocean biogeochemical features that we observe, and those are determined by a handful of processes, which we include into our model so that we can reproduce the observed features. There's no ecosystem model that is perfect. We do lots of trade-offs between complexity and simplicity and computational cost. Improving the models is still ongoing, of course. That's why we have jobs here. The global carbon cycle is present in many CMIT models, including CESM. And we're able to use observations of atmospheric CO2 on multiple timescales to constrain the models and see if they're doing well or poorly. And later tomorrow, there'll be practical notes for running with the carbon cycle in the LAN BGC breakout. If this is an interest in your research, then please be sure to attend the LAN BGC breakout, where we'll talk a little bit more about the practical aspects of running CESM with fully prognostic carbon. So I'll close there.
Really insightful and really great. This is one of my key passions, is the carbon cycle, and the ocean component. That's so valuable. It's the work that Keith has done. It's amazing. We have some time for questions. So if you've got questions, please come to the microphone and we'll let Keith answer them.
Is there any plans to include like macro zooplankton into the model because of like forams and pteropods and like their influence on like calcium carbonate?
There's an effort, actually, we're working on a configuration of the ocean biogeochemistry model now that includes a hierarchy of grazers. So we're not, for CESM3, we probably won't have a single grazer like we do in CESM2. I don't know if we'll do pteropods as one of them, but we do have microzooplankton. We have a cascade of zooplankton that are feeding on different size classes of phytoplankton, as well as on other zooplankton. So we are going to try to represent more of the food chain in CESM3, which we're still a ways from, but that is under development.
Thank you for the talk. I have a question. So I wondered, what is your opinion on iron fertilization and artificial upwelling in order to create this primary production in places where nutrients are limited, right? Do you think it's worth it to do it at large scale, or do you think the benefits are not sufficient to satisfy the risks? Maybe the increase of acidity in the ocean and so on?
I don't think we know yet. There's lots of questions regarding that. One is how much actual carbon is being sequestered to the depths. When we do fertilization, iron fertilization in regions where iron is below enough levels to support productivity, it certainly can increase productivity locally and lead to some sequestration of carbon, but some of the efficiency of that is poorly constrained, and it depends a lot upon where that iron fertilization happens. And I think an open question is, if you hadn't done that fertilization, would that carbon have just been sequestered somewhere downstream? And so being able to control for that's hard to control in the experiments. And there's modeling work where this is happening. And I think there's a lot of open questions about how effective it truly is for sequestering carbon, as well as what are potential negative side effects of doing that. And I think that's still an open question. So I'm a bit skeptical myself that it is about its promise for long-term sequestration of carbon. But I think there's a lot of that's my personal opinion, but there are a lot of open questions.
Okay, thank you.
Sure.
Can I just follow up on that? So if you were to be, say, like farming kelp for biofuel production, where you would be extracting it rather than worrying about the ecosystems being the sequestration, would that change the equation?
Well, I guess it depends upon what happens to the kelp once you grow it.
Right.
It would be consuming the carbon, removing it, but then what happens to the kelp down later? Right. And are you stealing the nutrients from downstream? Yeah. That there's the street. If you consume the nutrients here, then the critters that are downstream are not going to have the nutrients. And so there might have been, by consuming it with the kelp agriculture, aquaculture, you might just be moving where that biological productivity is happening.
Fabulous.
Matt Long at NCAR is actually involved with some projects looking at things like this, kelp harvesting and sargassum as well.
Fabulous. Thank you.
Hey, that was really interesting.
Thank
I
you.
have like a three-part question. So
Okay.
I study nitrogen fixation on land, but know shockingly little about it in oceans. So I was wondering if you could say anything about the kind of pattern spatially and temporally where it's happening the most. What controls it? Like, is it nitrogen deficiency or temperature or something else? And then how the model does that representing those patterns.
Sure, I'll try to tackle those. The nitrogen fixation in the ocean is performed by a handful, by relatively few autotrophs. Not many can do this nitrogen fixation. And one example of the class of characters is called diazotrophs in the ocean. The process of breaking down that dissolved N2 gas is just from the chemistry side of that, requires an enormous amount of energy to break the triple bond of the N2 molecule. And so that energy requirement to do that is fueled, I think, by iron. The critters that can do the nitrogen fixation have a high requirement for iron. And so that is one of the key regulations on where nitrogen fixation happens is where you have sufficient iron for the diazotrophs to thrive.
Is that because iron's needed for nitrogenase in the?
That's beyond my,
Yeah.
I don't actually know the biology. All I know is that the diazotrophs have a high requirement for iron. I don't know the more specifics about the biological aspects of why they require iron. They also tend to only thrive in warm regions. And so the nitrogen fixation in the ocean primarily happens at intermediate and low latitudes where there's lots of iron. And that's primarily where you have nitrogen fixation because of the requirements of the diazotrophs, which I have no idea about nitrogen fixation on the land. So I can't contrast it to the land.
Talk about that later. So the model doesn't represent iron. So is it able to capture this pattern?
Well, we do represent iron in
you do?
CESM. We have a sophisticated representation of iron in CESM. And so, and we have explicitly a diazotroph class of phytoplankton that do this nitrogen fixation. So this is included in the CESM ocean biogeochemistry model. And we're able to reproduce observed patterns of nitrogen fixation.
Thank you.
Sure.
Great talk. I have a small question. So based upon a 2018 Moore paper, we found that the fishery will decline in the simulation until 2300, the science paper in 2018. So do you think when you will include the flow of nutrient from land to atmosphere, there will be similar result expectation, or there is like some changed pattern that you are expecting? Because you said you're still working on the fishery model in the ocean part.
That's a great question. And I think we'll have to do the experiment to find out. I don't have an intuition about that's so many things can change.
It was a really, really fascinating talk. I have once you improve the biogeochemical the cycle in the model, so do you think the SST biases in the model will improve, or you have any idea how the cold or warm SST bias can be linked to the cycles?
The ocean biogeochemistry does feed back onto the temperature patterns of the ocean, primarily through chlorophyll in the ocean changes the how deep shortwave penetrates into the ocean. If there's lots of chlorophyll, then that shortwave is trapped closer to the surface. If there's no chlorophyll present, then the shortwave tends to get deeper. And so the solar heating, the depth profile of that changes depending upon the presence of chlorophyll. Um, but the way in our experience, this impact of biology on SSTs is sort of a second order effect compared to the radiative forcing that we get from the atmosphere, as well as the circulation and mixing that are in the ocean. So I wouldn't expect it. I would expect that the physical parameterizations has a first order effect on the SST properties. And also the resolution of the model has a first order effect in some regions. And I would think that the biology, in our experience, the biology, it does have an effect, but it's
Not
sort of
as
lower
big.
on the, it's a smaller effect than the other properties.
Okay, thank you.
Sure.
Great. Well, so that's all the questions for now. Thank Keith and Peter for these two great talks this morning.